Marianne is professor at the Department of Pharmacological Sciences and the director of the Mountain Center of Bioinformatics at the Icahn School of Medicine. And he's, of course, in the LINKS program. He's also part of the IEG program and the NIH Data Commons. And he's going to talk about his recent work towards mining 10 million signatures.
Thank you, Stefan, and thank you for hosting this great symposium. You've done an awesome job the first time in this round. It also looks like really nicely organized and it's a welcoming environment. And also it's great to have all the lab members here. And this presentation is also sort of like to give them credit for their work. So the LINKS program, the main paper that started LINKS is this connectivity map paper by the group at the Broad with Todd Valov, where they took several human cancer cell lines and then treated them with almost all of the FDA-approved drugs in different concentrations using microarrays and measured gene expression after 6 hours. And to create this resource where you can now come with your own signature of, let's say if you did some experiments in a mouse or in cell lines where you perturb the cells with a drug or knock down a gene, then you can find small molecules that either mimic your signature or reverse your signature, and all of you know about this. And so this is initially was done with microarrays. It was very expensive, and it only included, almost all of the FDA-approved drugs, but it's still a limited set of about 1,000 drugs. So to scale it up, the Broad came up with this L1000 technology that all of you have probably heard of. And the idea was that we can now do it with much higher throughput, very low cost. You only measure 978 genes, but those genes are carefully selected to be the most orthogonal genes where you can predict the expression of the rest of the genes. And it was shown in several benchmarks that it performs as well as microarrays. So very recently, the Broad team, the LINKS Broad team, published their first L1000 paper. And in the paper, they described signatures for about 20,000 small molecules and 5,000 also functional gain-of-function genes. And they divided the data into a reference set, which is called Touchstone, and a discovery set that include most of the small molecules. And also in their paper that they made an attempt to visualize the data. But data visualization is really challenging when you have so many data points. So they used T-SNE and they visualized the Touchstone reference side of the data and then projected on that reference set this particular set of 147 antibacterials. So in general, on the orange dots, there are about 2,000 points. And in those 2,000 points, the drugs are consolidated. So they have signatures of different cell types and different concentrations and time points. They're all consolidated to a specific perturbation, either a gene knockout, overexpression, or a small molecule. This data, we had access to this data early. The data was also made available publicly before publication. And the way we processed the data, first we focused on the chemical perturbation data, and that's about 200,000 conditions, different conditions that were profiled. And we processed the data and decided to basically filter the data to only include those signatures that were most consistent and significant. And then this resulted in about 4,000 drugs, 63 cell lines, various time points, and then 17,000 points are signatures for those drugs, different combinations and cell lines, clustered using this alternative approach that is a force-directed layout. And this is the work of Zichan, who's here. You can talk to him also about this, and that's his poster is about this data visualization. and tool that is available online called L1000 Fireworks Display. And the exciting thing about this tool is that you can really see the power of the L1000 data and in general the LINKS program as a whole. So here the coloring of this map is by mechanisms of action. And you can see that you have very clear clusters of small molecules that are clustered based on the mechanisms of action. And the gray points are small molecules that do not have known mechanisms of actions. So just by eye, you can already see that there are so many small molecules that don't have known function that can already be categorized as being either HDAC inhibitors, which are the green cluster here, tubulin, disruptive compounds that are in purple here, and there is also that cluster at the corner which has very toxic compounds that include CDK inhibitors and topoisomerase inhibitors. So now we have all of those novel compounds without known function that can be directly classified as having new mechanisms of action. So we can now project other types of coloring on this map, and this is coloring by batch. And one of the challenges with the LINKS data and in general when you start combining gene expression data or other types of data from many experiments is to remove this batch effect and to apply this method that was already developed to remove this batch effect. So now you can see that the various batches are all mixed together. This is coloring of the map by clustering. So this is a k-mean clustering type of coloring. So you can see that we can get very distinct clusters of small molecules signatures and the signatures that they produce. This is coloring by cell type. So you can see that some clusters are very cell type specific. So some of those signatures only turn on the genes, the same genes in some cell types. And this is, let's say, the green band that goes up, or this orange and purple band that explodes from the center. But some of those clusters have many cell types. So that means that for those drugs and for those signatures, it doesn't matter what cell type you use in the experiment, the same genes go up and down. So it's targeting the same pathways. So this is visualization by the phase of the drugs, like what are they already marketed drugs, are they FDA approved drugs, or are they in clinical trials? There's some withdrawn drugs there. And all the gray ones are drugs that haven't been tested in humans yet. We also, surprisingly, get some clustering by the molecular structure of the drug. So if we project the different scaffolds of those drugs, we can see that there is some structure expression relationship for some of those red spots at the top. Those are protein synthesis inhibitors that all share the same scaffold. So now we can also go to a medicinal chemist like Stefan, and we can give him the list of drugs or the scaffold for those drugs, and we can also design novel molecules that we will know likely to cause that desired effect. And I think the most exciting part of it is that we are able to link this map to the Mount Sinai electronic medical records. And here we are coloring the points for those drugs and have data about usage in the clinic in real time at Mount Sinai. And we can now zoom in to this graph. So if you go to this URL, you can interact with this resource. And we can zoom in, and the nodes or the points that have color on them, they have data from the Mount Sinai electronic medical records. And if you click on one of them, you get a signature report. So every point on this map is referring to a signature. So that means that this is some experiment, L1000 experiments, where a specific cell line was used and was treated with that small molecule compound in that specific dose, and gene expression was measured at a specific time point. And then you can get the up and down differentially expressed genes. You can click on a link to enricher to do enrichment analysis on those up and down differentially expressed genes. You also have a lot of metadata about the small molecule with external links, including links to the links data portal and iLinks, which are developed by the other teams from the DCIC. And here is the data from the Mount Sinai EHR. So this is a diagnosis. So this is the various ICD-9 codes of what is the drug. The drug is actually used in the clinic. And also we have collected co-prescribed drugs. So for every drug, typically patients are given multiple prescription and multiple drugs and take multiple drugs at the same time. So these are the most co-prescribed drug for this particular topoisomerase inhibitor that is used mostly for leukemias. And this is the distribution of patients that are taking the drug. So this is the age distribution of those that are prescribed this drug. So one thing that you can do, besides actually by eye, identifying what is the mechanism of action of the drug, you can do it more systematically using machine learning. And we can predict the mechanisms of actions for all of those drugs on the map. And for this particular drug, it's already known that it is a topoisomerase inhibitor, but we can also put it in this predictive model, and it does predict that it is a topoisomerase inhibitor. But some drugs with known mechanisms of action, if you put it in this predictive model, the predictive model predicts them to have different types of mechanisms of actions that are a little bit unexpected. For example, for this drug, which is a known histamine receptor antagonist, it also predicts that it is a dopamine receptor antagonist and a calcium channel blocker. So how do you set up those predictive models? So we can take properties of drugs and small molecules, and this is the morphological features. So this is about 812 features. And then we also took the chemical structures of the drugs and converted them to a binary string. And then we have also the 978 landmark genes as features. And then you can build your classifier. It can be neural networks, or it can be random forest. I think in our case we used random forest classifier. And then on what we want to predict is adverse events. And what we showed in this paper is that if you combine those features from those various resources, you can get an improved prediction of side effects. And the same way it was done, we sort of like realized it later, we can also predict mechanisms of action and potential targets. And one of the things that we are trying to do at the DCIC is also create our own signatures. So even though we are dry and we don't do experiments, we are doing a lot of mining of signatures from the public domain. And this sort of like started through this Coursera course that we were teaching when very early times of Coursera. And the exciting thing about those courses is that there were tons of people that are coming to those classes. And for this particular session of this course, 21,000 people registered. 10,000, half of them actually showed up to the course, so people just registered, but they did not bother even like to come to the course. And then 388 paid $50 to get a certificate. Half of the 10,000 never heard of Mount Sinai, and 200 of them actually were from Mount Sinai. So this is interesting statistics about the participants of that course. So for those people that attended the course, we offered to participate voluntarily, not as a part of the course, in this crowdsourcing project, where we asked them to use this Chrome extension called Geo2Enricher to go to the Geo database and mark control versus treatment samples to extract signatures. So this is the geo already built in way that you can use to mark, let's say, control samples and perturbation samples for estrogen in MCF7 cells. It's like a typical study. And then once you make that selection, you are presented with this geo page, but now it has this embedded line in the page that the Chrome extension adds. And by clicking on this button, we can now send those files, those GSM samples from Geo to our server and compute a signature, compute the differentially expressed genes in the control versus the treatment samples. So together, the students in the course, they extracted 2,000 single gene perturbations, about 1,300 drug toxin perturbations, and then also comparing disease versus normal tissues. And now we have this nice database that has all of those signatures that were manually extracted from literature. And those signatures are served on this website called CRE. And people are using those signatures for various things. The thing that was a disadvantage of that resource is that it's all microarrays, all technology that is now replaced by RNA-seq. The challenge of RNA-seq is that you can't really do it with RNA-seq because the data on Geo is in raw form. So, and it's some of it that is processed is not processed uniformly. So there is no way to download those sample files and compute a signature. So Alex Lackman developed this really efficient pipeline to systematically process all of the data from Geo. And the key thing about this pipeline is that it's very cheap. So it costs less than a cent per sample. So to process all of Geo is about $1,500. There also the key thing that he's using this aligner called Callisto. So now you have all of this data processed and served on Geo through a Chrome extension that is shown here. So now you have embedded with a Chrome extension access to all of those processed files. So one of the things that Alex also did for this project, he visualized all these samples using a 3D t-SNE visualization. Also what we've done using the core expression data from collecting all of the samples, we were able to predict protein-protein interactions and functions for genes using a relatively simple algorithm. So we combined the prior knowledge about protein-protein interactions with this core expression data to make predictions about understudied proteins and genes and assign to them protein interactions and molecular functions like GO terms, memberships in pathways, and human phenotypes. So all of this data is served on this website called Arches4, where you can put in any type of term to identify subset of samples that then you can download and process and work. So I mentioned this Chrome extension, and this Chrome extension was developed by Dennis Torre together with Alex. And Dennis is now building another Chrome extension that is sort of like the next version of this Geo2Enricher Chrome extension that was used by the students of Coursera to extract those signatures from Geo. And this Chrome extension will have a button next to each of those Geo studied entries, and those are going to be RNA-seq studies. And once you click on this button, you will launch a Jupyter notebook as a report that contains analysis with multiple tools. So our signature tool, our most famous or most successful effort so far has been this program called Enricher, and this is a relatively simple enrichment analysis tool. And the advantage of Enricher over other systems is the breadth of the types of data that we have processed and included in this resource. And currently there are 234 terms that are gene sets, annotated gene sets, that people can query their own gene sets to identify potential functions. And this tool has been widely used, so almost 9 million submissions of gene sets have been already accumulated in this resource. So people come in with their gene set and then they submit it for analysis, or they can use an API to submit genes for this enrichment analysis. So this is, if you click on the number of submissions, you get this visualization of how many lists are submitted every day. And what we are trying to understand next is like how people actually using this tool, and what data they're submitting to Enricher. And potentially one of the things that we try to do, we're not going to make their submission public, but we can somehow use all of this accumulated knowledge that is submitted to the tool and potentially learn some biology just from those submitted lists. So in general, surprisingly, most of the lists that are submitted to Enricher are actually short. They're containing less than, I guess, 100, 200 genes. And then there is this interesting bump. So there is a bunch of genes that are submitted very rarely, but there is a bump of the frequency of genes that are submitted, and there are some genes that are submitted very frequently. So this is it's like a long tail of submitted genes. And if we look at it more carefully, and what Alex did, he divided the lists to long lists versus short lists. And we can see that in the short list, there is overrepresentation of those well-studied genes. We also analyzed the gene sets, the annotated gene sets that are in Enricher, and there are biases also with those lists. So those 200,000 annotated genes that people are searching against also contain biases, and this is the work that the idea here is that if you cluster the most common genes from each library, you get clustering by library. So that means that depending on how you acquired your data, whether it's from ChIP-seq, RNA-seq, microarrays, whether it's like a pathway database, those type of data acquisition methods, they have a bias. They're going to have genes that always show up more frequently than others. So all of those biases have to be considered when you are doing those experiments, when you are trying to query your signatures against other signatures. So another app that is up and coming is still in development. It's a web implementation of an algorithm or a pipeline that we have developed many years ago. And the idea is that you identify the signaling pathway upstream of a signature. So you put in your differentially expressed set of genes, and then you automatically get this pathway that has the transcription factors at the bottom. So the process does enrichment analysis for transcription factor first, and then we connect those transcription factors through protein-protein interactions to build a subnetwork, and then we apply kinase enrichment analysis on this pathway to identify the kinases that are most enriched for that subnetwork. So the idea is that when you do gene expression analysis, you really don't know how those genes are changing at the protein level, what are the pathways that are activated upstream or downstream, and in this way you can recover the pathways. Using links data and other data where kinase was perturbed either by a drug or by a knockdown, we can now see, can we recover the pathway or the actual kinase that was perturbed by looking at the differentially expressed genes and then running this pipeline to recover the kinases. So immediately it works much better than random, but we can improve it. All of the parameters of the pipeline are described as a binary string, and then using genetic algorithms, we're trying to optimize the parameters by going through the cycle of validation and seeing whether we can somehow increase the prioritization of those right kinases or right drugs that are affecting those kinases. So there are various ways to perform this enrichment analysis for transcription factors, and there are multiple different libraries that are have a transcription factor as the term, and then the target genes or the co-expressed genes with the transcription factors are in the members of the library. And what Ali is trying to do is try to optimize and benchmark those libraries. So you also have data from transcription factor knockdown or overexpression, and now you can see how much we can recover the perturbed transcription factor. And this plot shows that if you combine multiple libraries together, you can get more accurate prediction of the right transcription factors. So surprisingly, there are various sources for drug target associations, and they don't agree with each other. So the community as a whole don't really have consensus of what are the targets for drugs. Benchmarking those resources, trying to see which of those databases is actually better, which one has a more accurate set of drug target interactions. And the way he did it, he used the links data by looking at how much the drug cause the same signatures, or how much there is correlation between like the drug-drug network that is created from the links data, the drug-drug network that is created based on the targets of those drugs. So it's the idea that we can organize knowledge in various ways. So the drug target type of association is from this node here to the central node. So there are connections or bipartite connections between drugs and genes. But in general, we identified 5 major types of objects that could be connected, which are cell lines, tissues, disease, phenotypes, adverse events. The genes and proteins are sort of like in the center, and those could be targets. And then you have gene sets, modules, pathways, and then small molecules. And it's not only connections, so all of those things can be connected to each other, but also you can make connections within each of those. So protein-protein interactions, for example, would be a connection within the gene-protein targets. And you write some scripts to convert all of this data to those various comfortably used normalized data structures that people can then use for machine learning and data integration. So I think the most important activity that we actually do at the DCIC is the outreach. And one of the major activity for the outreach is this summer research program. And we had like really great success with this summer program. And the applicants that submitted to participate in this program are awesome. Like we get mostly computer science majors. And if you look at their GPA, so the average GPA is 3.6 almost, and many of them are from Ivy League schools. And those are people that haven't really decided what they want to do with their career, and we really had converted a lot of them to be really interested in biology, bioinformatics, big data science in our field. So it's really nice to see how we convert those young minds to be interested in links, to be interested in big data and applications in medicine. And that's it.